the increase of absorptive transport by 20 micromoles quercetin in K2 cells is comparable to 100 micromoles verapamil, indicating that quercetin is a fairly potent PGP inhibitor. Wow. So 1/5 of the quercetin is all it takes compared to verapamil, which we've talked about before. Remember the rat study where they gave ivermectin with verapamil and it showed much more profound effects for the sperm quality. So is the same thing happening for people taking quercetin with ivermectin? This needs to be investigated. Quote, this study shows that Natto K2 inhibits PGP in vitro. Natto K2 inhibits PGP in vitro. The paper says, quote, special attention should be paid to Natto K2 due to low IC50 value and low IC50 over IC25 ratio. So IC stands for the inhibition concentration. And then the number after is what concentration inhibits that percent. So IC50, what concentration inhibits 50%? IC25, what concentration inhibits 25% of the flux? So here's some really interesting findings from the paper. Look at this chart. What really jumps out is how little Natto K2 is required to inhibit 50%. Just 0.62 milligrams per milliliter. 0.62. Compare that to well-known PGP inhibitor verapamil, which takes 5.4 milligrams per milliliter to inhibit 50%. That's a huge difference. But what's really concerning is once quercetin is in there and they're exposed to this arsenic, there's next to no PGP relative to these other measurements. It goes way down. So usually when arsenic is added, we get this big spike in PGP. But when quercetin's in the mix, where's all the PGP? This is pretty remarkable. Not just that it seems like quercetin might reduce the amount of PGP, but it inhibits the normal response, the normal detox response with PGP, and it's gone. Where's the PGP spike? Quercetin, however, completely inhibited the increase of the P-glycoprotein synthesis caused by exposure to arsenite. Hey everybody, I want to cover some more information today about P-glycoprotein and ivermectin toxicity. Some of this I believe also applies to HCQ, hydroxychloroquine toxicity, given it's also a PGP substrate, but I really want to focus in on ivermectin. Previously we've discussed the role of P-glycoprotein in reducing ivermectin toxicity. And the way it works is it reduces the toxic pesticides exposure to our brains and testes, among other vital tissues. And the reason I bring up brain and testes is because it has a neurotoxic effect when it gets into our brain. So that's not good. And it seems in multiple animal studies to have negative fertility effects when it gets into the testes. So P-glycoprotein is a crucial protein at these barriers. So it's also in the small intestine, the liver, the kidney, and placenta. And it keeps toxic substances like ivermectin out of our tissues. The problem is some people don't have as much P-glycoprotein. And there's two reasons for this. Some people don't express as much because of their genetics. There are mutations of the MDR1 gene, which seems to correspond to PGP. So some people, because of their genetics, will produce significantly less PGP. There are also PGP inhibitors, which in a way can block these channels, block these pumps, and prevent the PGP from doing its job. For other substances, prevent our tissues, our cells from detoxing the PGP substrates. And what's also interesting, I just learned, it seems like PGP expression increases when exposed to toxins. And I'll show you what I'm basing this on in a second, but the body tries to mount a response, and it does so by using mRNA to express more P-glycoprotein. That is, unless you take herbal substances like quercetin, which interfere with that process, at least for arsenic. Now, in the prior article, we discussed how ivermectin, hydroxychloroquine, some statins are PGP inhibitors, P-glycoprotein inhibitors. And that means they weaken the PGP defenses against ivermectin at some very important tissues. So P-glycoprotein protects us from ivermectin, but there are inhibitors, which will allow it to more freely cross through, which will cause the ivermectin to not be able to be expelled from the cells as readily. Today we're going to cover two more PGP inhibitors, Quercetin and Natto, which have been pushed by some people who also promote ivermectin. Or we'll see some people pushing quercetin and others pushing ivermectin. But people are listening to all these videos, so I would not be surprised if people are on quercetin and or Natto and taking ivermectin. So I really want to reach those people and show you the facts that I've come across so you can take this into consideration. I want to go through a little bit from this last article, P glycoprotein deficiency. If you check out the link down below to my Substack, it'll have all this information for you. But what's very concerning is they measure this various different ways. Genetically predisposed animals and or the PHP inhibitor use to see the role of PGP in ivermectin toxicity. And it seems to be very important to minimizing the ivermectin toxicity. So I think it's reasonable to conclude, on the basis of a preponderance of evidence, that P glycoprotein protects us from ivermectin toxicity. And by inhibiting somebody's PGP and then giving them ivermectin, that's a way to poison somebody intentionally. So we looked at this example where they had the knockout mice, the MDR1A negative knockout mice. So much less PGP. And what we see is the dose required to kill these rats is about 100 times less than the more normal rats with the adequate PGP. In fact, they often test dogs before they give them ivermectin to make sure they don't have this gene mutation. So these people pushing ivermectin on human beings aren't even doing the same courtesy many vets do for their dogs. Though interestingly, sometimes for these studies for ivermectin's effectiveness against COVID-19, they will exclude people who have this mutation. So here's one of these studies. It says, we performed sequence analysis of the MDR1/ABC AB1 gene in patients included in the study group. Patients with mutations were excluded from the study, and ivermectin treatment was not continued. So for all of these ivermectin pushers who call this safe, tell us what's the difference for somebody with and without this MDR1 mutation. How much more toxic is it to the people with this mutation? If you don't know, then how dare you call it safe? And also, what about the drug interactions? What's the safety for somebody on quercetin? What's the safety on somebody on Natto? Etc., etc. There are so many unknowns not being relayed to the unsuspecting masses. And we see in this chart how much more ivermectin accumulates in various tissues with and without this gene mutation, which corresponds to reduced PGP expression. And what we see is 87 times more ivermectin accumulating in the brains of the MDR1A negative polymorphism, and 7.45 times more ivermectin in the testes when they have this genetic predisposition, limiting the amount of P glycoprotein. We have this other study that shows a 99% reduction versus the normal in terms of the lethal dose required to kill 50% of these rats they looked at in a different study, if they had the negative polymorphism. If they had the positive negative polymorphism, it was a reduction of 53% in the ivermectin required to kill 50% of the rats. And here's a really interesting depiction of the PGP expression using Western blot on the brain and the testes for each of the three polymorphisms. We see substantially more PGP for positive, substantially less for negative, almost none, and somewhere in between for positive polymorphism. And there's a huge list of 332 P glycoproteins on DrugBank. Verapamil is one of the most widely known. But we can also see quercetin. We can also see ivermectin itself is a potent PGP inhibitor, which calls into question the safety of cumulative dosing. I think there's questions in one-off dosing. There was the 13-year-old boy who was put into a coma by one pill, and they said it's because he had a predisposition genetically. Hydroxychloroquine and chloroquine are also PGP inhibitors. And I've recently found that Natto K2 also inhibits PGP, it seems. Now, one more study before we dive into the quercetin and the Natto K2. This is really interesting. This uses verapamil along with ivermectin. And we see these profound effects of a PGP inhibitor being used in tandem with ivermectin. Huge drop in sperm count. Down 12% with just ivermectin, but down 46% when they paired it with the PGP inhibitor verapamil. Sperm abnormality, when these two are paired, went up 59%. Sperm motility, the percent swimming sperm, down 23% when ivermectin was administered with the PGP inhibitor. This is major. This is absolutely major. So why are we hearing people pushing quercetin, while others, sometimes they're the same people, are pushing ivermectin? Just a coincidence? Why are we hearing people push Natto K2, while others are pushing ivermectin? Just a coincidence? So let's get back to the main article here. Before we dive in, I want to cover some key concepts. A PGP substrate is something that these PGP pumps expel from the cell. So when something's called a PGP substrate, that means the cells can get it out using PGP. That's my understanding. Now, there might be other ways that they can detox themselves, but the term PGP substrate means that the P-glycoprotein deals in these substances and will shuttle it out of the cells. For example, here we see digoxin, which we'll talk about in a second. So PGP inhibitors stop these channels, stop these pumps from expelling the PGP substrates from the tissues, from the cells. So let's talk about quercetin and natto. So let me start with some terminology with nattokinase versus vitamin K2 or Natto K2. It's a little confusing when they sell supplements called nattokinase, whether it includes Natto K2 or whether it's been removed. And then there's the question of, for the products that say it's removed, do they remove 100%? Do they remove every single molecule? Or is there still some mixed in, but not a lot? Well, according to this article here, it says K2 is found in nattokinase. Various nattokinase supplements contain vitamin K2. So the study that we're going to look at talks about Natto K2, not nattokinase. So I just want to make this very clear. Now, I think nattokinase has some other questions that should be asked. And I haven't seen a study that says whether or not nattokinase is a PGP inhibitor. All I was able to find is that Natto K2 is a potent PGP inhibitor. So I was looking around. On Amazon, you can buy Natto K2 separately from nattokinase. The labels look very different. But I don't know which of these nattokinase products have K2 in it. And I also don't know what nattokinase does by itself. They say it's a protease, which means it breaks down proteins. That's my understanding. They say nattokinase comes from a Japanese food called natto. So to produce natto, which is a Japanese food, according to this article, they boil soybeans, and then they add this bacteria, Bacillus natto. And apparently the bacteria produces the enzyme. And they say nattokinase is a serine protease, which are enzymes that cleave peptide bonds in proteins. And I see people pushing this for spike protein detox. And my question is, what about all the other proteins in our body? If they're taking these proteases in an attempt to break down their spike proteins, what's going to happen to all the other vital proteins in our bodies? These are just questions I have. Like, what about the P-glycoprotein that we've been talking so much about? So I've seen that there hasn't been much studying about nattokinase, but I'd love to see the safety studies. I'd love to see the combination studies with ivermectin and nattokinase, or especially with Natto K2. But we can look at the studies that we do have and try to extrapolate. This is a really interesting paper linked in the article, Effects of Herbal Remedies Frequently Used by Cancer Patients, by Engdahl and Nilsson. So the researchers looked at Natto K2's inhibition of digoxin flux. So the researchers looked at Natto K2, along with other substances like verapamil and mistletoe, and they looked at how much it took of these substances to inhibit digoxin flux. So they used some cells, I believe, from the small intestine of a cancer patient, called K2 cell line. And again, digoxin is a PGP substrate. So if you block the PGP pumps, the flux is going to go down, and your cells are going to accumulate this stuff. That's the idea. And a lot of this research is because these cancer drugs get expelled from vital tissues. And so Big Pharma and the other drug makers are trying to shut down the body's detox mechanism in order to get these toxic substances to stay in the tissues. But look at this chart. This is really stunning. On the x-axis is how many milligrams per milliliter of Natto K2. And on the y-axis is the flux of digoxin as a percentage of control. And we see as the Natto K2 increases, the flux gets cut off. So at the end of this drop-off, digoxin is practically not even flowing. That's huge. Quote, this study shows that Natto K2 inhibits PGP in vitro. Natto K2 inhibits PGP in vitro. The paper says, quote, special attention should be paid to Natto K2 due to low IC50 value and a low IC50 over IC25 ratio. So IC stands for the inhibition concentration. And then the number after is what concentration inhibits that percent. So IC50, what concentration inhibits 50%? IC25, what concentration inhibits 25% of the flow, of the flux? So here's some really interesting findings from the paper. Look at this chart. What really jumps out is how little Natto K2 is required to inhibit 50%. Just 0.62 milligrams per milliliter. 0.62. Compare that to well-known PGP inhibitor verapamil, which takes 5.4 milligrams per milliliter to inhibit 50%. That's a huge difference. Much, much less Natto K2 is required. What about for 25% inhibition? Well, compared to verapamil's 0.54, Natto K2 only takes 0.2 milligrams per milliliter. And this stuff is readily available on the internet, on Amazon, etc. So we have to start thinking in combination. It's not just the safety of any one drug or any one herbal remedy. There's some big problems that can happen when you start combining substances. And what's also interesting is verapamil, it takes 10 times the amount to inhibit 50% than to inhibit 25%. You can see that over here on the right, the ratio of 10. For Natto K2, it only takes 3.1 times the amount to inhibit 25% to inhibit 50% of the flux. So a very small amount does a lot with Natto K2 versus PGP. That's pretty amazing. So this is interesting. This is a pharmacist who's a pretty big personality when it comes to long COVID on Twitter. So putting aside the whole concept of long COVID, this person is probably responsible for getting a lot of people to take Natto K2. They post alleged survey results saying, oh look, what topped the list for long COVID patients? Natto K2. So I don't know her intentions or anything, but what's interesting is she came out later after telling a lot of people trying to get relief for their, quote, long COVID to take Natto K2. Well, a few months ago, she came out. This was November of 22. And she asks, drug interactions with Natto K2? She says, you may notice I mentioned P-glycoprotein in quite a few of the interactions. In my readings, I found in vitro studies showing that Natto K2 inhibits this protein. And I believe this is the study we just looked at. P-glycoprotein is an efflux pump, which means that it essentially pumps drugs out, thus reducing bioavailability. Or you could look at this as detoxing you, right? PGP appears in the blood-brain barrier, small intestine, kidney, liver, and tumor cells. So they add that because they have this issue getting chemo drugs into tumor cells. But they don't tell you about all these other cells in your body. They're like, oh, look what happens when we take these cancer cells and we inhibit their PGP. We can get the drugs in, without considering the complex system that is the human body. She continues, it limits drug absorption in these tissues. If a substance inhibits PGP, like Natto K2, it may increase the absorption of certain drugs, called PGP substrates, taken with it. So there's a lot of PGP substrates. So I thought this was interesting coming from somebody who, in a way, is giving a lot of people information leading them to take Natto K2. To come out and say, well, here's some information about the drug interactions, P-glycoprotein, the PGP substrates, and the PGP inhibitors. Now, she doesn't really talk about ivermectin in this write-up, because that side of the aisle isn't guzzling this stuff down. But ivermectin is a key concern when you're inhibiting your PGP. All right, let's continue to quercetin. It was much easier to find information about quercetin and its effect on PGP inhibition. In a 2005 paper called Inhibition of P-glycoprotein Function and Expression by Camphorol and Quercetin by Limtrochel et al., the researchers examined quercetin's effect on the influx of the PGP substrate RH123, and they used the KBV1 human cancer cell line. All of these papers are linked in full on my article, timtruth.substack.com. Link down below. So these are two very interesting and concerning charts. So we have the P-glycoprotein substrate, RH123. But we should also think about the other PGP substrates like ivermectin, which PGP seems to help us detoxify from. Well, let's look at the accumulation and the retention of this PGP substrate. Well, what we see is the control level at 100%. Everything's normalized based on the control. That's in yellow. And then we have the data points in red with the trend line. So we see as the quercetin increases on the x-axis, the amount of both accumulation and retention of the PGP substrate RH123 increases. And these are big increases in my book, especially this bottom chart of the retention. Quercetin is measured in micromoles, and you can see on the right end of the spectrum at 200 micromoles, the accumulation is 112.2% of the control, and the retention of RH123 is 149.1% of the control. So up 49% in the retention of the RH123. Then there's this diagram from the paper as well. We can see more Western blots showing the control has way more PGP than once the quercetin is introduced. And this is also in column chart format down below. We have the control with no quercetin on the left, and everything else is normalized to that being 100%. Then with 10 micromoles of quercetin, it goes down to 80%. And with 30 micromoles, it goes down even further. So a dose-dependent reduction in P-glycoprotein levels. Here's a quote from the paper. The PGP level in KBV1 cells were decreased markedly in the presence of 10 and 30 micromoles quercetin for a period of 48 hours in a dose-dependent manner. Quercetin at a concentration of 30 micromoles significantly decreased the PGP expression in KBV1 cells. The next paper looks at two hamster ovary cell lines, including one that's called a multi-drug resistant cell line. And this is interesting because they call one of the genes that's correlated with PGP expression MDR1, which is multi-drug resistance 1. So presumably this CHRC5, multi-drug resistant hamster ovary cell line, is really fortified with this PGP protection. So this is a 1997 paper called The Effect of Quercetin on Hosht 33342 Transport by Purified and Reconstituted P-glycoprotein by Shapiro and Ling. And the researchers took this quercetin product and they evaluated its effects on the transport of the PGP substrate called Hosht 33342. Quote, quercetin reduced the rate and extent of the decrease of Hosht 33342 fluorescence of both cell lines. Quote, our preliminary immunocytofluorimetry studies indicate that quercetin reduces the amount of PGP. And let's look at these two charts I made from the paper's data. So on the top is the CHRC5, which is the multi-drug resistant hamster ovary cell line. And down below is another hamster ovary cell line called OXB1. And we see this marked increase in fluorescence when the in vitro cells were given 100 micromoles of quercetin. So the idea is if the quercetin wasn't there, this PGP substrate would be effluxed from the cells, and they'd no longer be fluorescent, because the fluorescence corresponds with the presence of this PGP substrate. So the blue line's the control, and then the red line shows you that with quercetin, the cells are not able to detox this PGP substrate as efficiently, indicating prolonged exposure. And a lot of this research, like I said earlier, is focused on, well, if we can get cancer drugs to stay in the cells, it'll be more impactful. So that's a whole other side of this discussion as to whether people should be taking those cancer drugs. We're not going to get into that in this video. But what's fascinating to me is the implications that this has on people who are taking ivermectin, who are depleting their PGP army with various over-the-counter drugs. And to me, it seems a little suspicious that various over-the-counter drugs that inhibit PGP so much are being pushed in tandem with other people pushing ivermectin. Here's another quote from the paper. Treatment of the cells with 100 micromoles of quercetin during Host 33342 uptake resulted in a reduction of Host 33342 fluorescence in OXB1 cells. Increased Host 33342 uptake by CHRC5 cells suggests a possible inhibition of P-glycoprotein by quercetin. Now remember, this is like 25 years ago, this study, and this has been fleshed out with other studies since. So this chart is interesting as well. It looks at the percentage of the control ATPase activity with 25 micromoles and 50 micromoles of quercetin. And then we have at the very top the control line at 100%. So everything is what percentage of the control levels. So we see the ATPase activity goes down with 25 micromoles and then goes down even more, substantially more, with 50 micromoles of quercetin. So why is this important? Well, P-glycoprotein is an ATP-dependent efflux pump. So P-glycoprotein's function depends on ATP. Let's look at 3 more quotes from this paper. Quercetin inhibited ATP hydrolysis by P-glycoprotein. Quercetin inhibited the ATPase activity of P-glycoprotein, impairing its transport function. Quote, since drug transport by P-glycoprotein is dependent upon ATP hydrolysis, we checked for the inhibition of the ATPase activity of P-glycoprotein by quercetin. Indeed, quercetin inhibits the ATPase activity of P-glycoprotein. And that's what we covered in this prior chart. Okay, but it gets more interesting. Here's a paper from 10 years after. They did in vivo and in vitro studies. They used Wistar rats. So this was a 2008 paper called Preclinical Evidence for Altered Absorption and Biliary Excretion of Irinotecan in combination with quercetin. Possible contribution of P-glycoprotein by Bombshell et al. And irinotecan, aka CPT11, is another PGP substrate. Listen to this quote from the paper. The concomitant administration of naturally occurring dietary supplement quercetin significantly inhibited the P-glycoprotein both in vitro and in vivo. Quote, the amounts of irinotecan and SN38 eliminated in bile in control rats is reduced to almost half when treated with quercetin. So SN38 is a metabolite of irinotecan. So that's why they measure that as well. But these 2 charts, just for simplicity, just look at the CPT11 excreted in the bile. So bile is secreted by the liver and stored in the gallbladder. And they tried 2 modes of administration, both intravenous and oral. And for each of these charts, we have the control line well above the quercetin line. And the quercetin dose was 20 milligrams per kilogram. And we see much more is found in the bile when it's intravenously injected versus oral. So we see the addition of quercetin drastically changes how the body deals with this PGP substrate. And much less of it is found in the bile when quercetin is present, possibly indicating it's not being excreted, but rather absorbed, and the body's not detoxing it. So this is the first 10 hours, and we see plus 40% excreted in the bile in the intravenous mode of administration, and for the oral mode, 34% increase versus the quercetin level when no quercetin is administered. So the body is much more able to get rid of these substances, oftentimes toxic substances, without the quercetin present. So please keep this in mind. 2 more quotes from this paper. The increase of absorptive transport by 20 micromoles quercetin in K2 cells is comparable to 100 micromoles verapamil, indicating that quercetin is a fairly potent PGP inhibitor. Wow. So 1/5 of the quercetin is all it takes compared to verapamil, which we've talked about before. Remember the rat study where they gave ivermectin with verapamil, and it showed much more profound effects for the sperm quality. So is the same thing happening for people taking quercetin with ivermectin? This needs to be investigated. So 1/5 the verapamil for quercetin's PGP inhibitor, making it a fairly potent PGP inhibitor according to these authors. So 1/5. And one final quote, based on the results of our in vitro studies in K2 cells, quercetin is a potent inhibitor of P-glycoprotein. So Natto K2 and quercetin, at least in this K2 cell line, appear to be much more inhibitory for P-glycoprotein than verapamil, which we covered at the onset, when paired with ivermectin enhances the negative effects for the fertility metrics in rats. All right, so we have 2 more studies. The last one is a bombshell, so stick with me. The next one. So this is a 1994 paper called Quercetin Potentiates the Effect of Adriamycin in a Multi-Drug Resistant MCF7 Human Breast Cancer Cell Line, P-glycoprotein as a possible target, by Scambia et al., where the researchers examined the PGP substrate RH123 and the increased levels of RH123 as the quercetin was increased. This apparently coincided with a lowered amount of immunoreactive PGP. So here's a chart with the data they collected. This is the percentage of RH123 fluorescent cells as quercetin increases. So on the x-axis, we have quercetin in micromoles, and we see the yellow line is at 18.2%. That's the control level. So in the control, without quercetin, 18.2% of the cells were RH123 fluorescent. Now, as soon as they started adding quercetin, this PGP inhibitor, we see the percentage of cells that are fluorescent explode. And this is dose dependent. As the dose is increased, more cells take on this RH123 and don't expel it, importantly. So at 1 micromole, it's 60.5%, which is a huge increase from the 18.2% control. And the divergence only gets larger. At 5 micromoles, it goes up to 77.3%. And at 10 micromoles, up to 83.6%. Wow. And here are 2 quotes from the paper. In a comparison of the curves of the cumulative frequencies of fluorescence intensities from the control and Q-treated cells, so quercetin-treated cells, it appeared that 10 micromoles lowered the amount of immunoreactive PGP. So it lowered the amount of immunoreactive PGP, P-glycoprotein. And remember, this was like 20 years ago. It says, at present, our results do not explain the mechanism by which quercetin modulates PGP activity and reverses MDR. So again, MDR, multi-drug resistance. So I don't want my multi-drug resistance being reversed, especially if I was taking ivermectin. It says a direct interaction between quercetin and PGP may be possible. Now, this last study, I think, is the most bombshell. And this was from way back in 1992. This paper is called Quercetin, a bioflavonoid, inhibits the increase of human multi-drug resistance gene, MDR1 expression, caused by arsenite. And they studied this in the HCPG human cell line, which is a cancer cell line. The researchers found that quercetin inhibits 2 very important things, both the increased MDR1 mRNA and, in turn, the increased P-glycoprotein synthesis. And this is usually seen when cells were exposed to arsenite, which, according to some cursory research, appears to be the most toxic form of arsenic. So they exposed these cells to a toxin, arsenic. And what usually happened, as you can see in this second column here, is the PGP spiked. And to me, that sounds proper. Like, oh, we need some more P-glycoprotein in here to help us shuttle out this toxin and detoxify. So we see on the left the control levels of PGP. So this is a Western blot analysis for PGP. So in column 1, we see the normal level, the control level. In column 2, we see the arsenic, and we see this big spike in the expression of PGP. Then for number 3, just quercetin. And what's interesting is we see this lowering of the PGP when quercetin is added. But what's really concerning is once quercetin is in there and they're exposed to this arsenic, there's practically no PGP. There's next to no PGP relative to these other measurements. It goes way down. So usually when arsenic is added, we get this big spike in PGP. But when quercetin's in the mix, where's all the PGP? This is pretty remarkable. Not just that it seems like quercetin might reduce the amount of PGP, but it inhibits the normal response, the normal detox response with PGP. and it's gone. Where's the PGP spike? I, for one, want my PGP to spike when there's a toxic PGP substrate in my body. I don't want it to go to zero. So this might be one of the most important studies I found. And here's the last quote that we'll cover. The exposure to arsenite caused a 2.2-fold increase in the P-glycoprotein synthesis. That's right here. That's 2.2 times. Quercetin slightly inhibited the constitutive synthesis of P-glycoprotein. Quercetin, however, completely inhibited the increase of the P-glycoprotein synthesis caused by exposure to arsenite. This all makes me feel absolutely sick, because I know what information's flying around and what people are being pushed into taking. Why was quercetin pushed on people? Why was ivermectin pushed on people? Why is Natto pushed on people? These are the questions we need to ask. And I refer you to timtruth.substack.com, where you can read all these articles and the corresponding video walkthroughs for my ivermectin and P-glycoprotein series. More to come, by the way. And also check out the playlist tab on timtruth.com. There's a free playlist with all my ivermectin toxicity videos if you want to get caught up. I think this is Bombshell. I think this is huge. And I think it might signify a mass betrayal. Think about the effects that verapamil had with ivermectin in the rat studies. And then think about what we just learned about quercetin having, at least on some cell lines, an even more profound effect on PGP inhibition. And then we saw Natto K2, also a very profound, seemingly more profound than verapamil, which they looked at as well, effect on PGP inhibition. And then we can cross-reference that to the ivermectin toxicity and the importance of PGP. Please connect these dots, people. Are these binary weapons designed to be extra poisonous and have negative fertility effects when taken in tandem? If so, what are the motives? What's the intent of the people pushing the different components of the binary weapon?